Hey, Teacher Thoughts here, coming at you with the long-awaited video where I bring in the science teacher. This is one of my former coworkers. We work together here at the local high school. Both of us have backgrounds that are a little bit different. My background, not only am I a teacher, but I was a tradesperson. So I worked in manufacturing and then spent my entire life in the nursery industry. Our science teacher, however, had a little bit different of a life path and has some credentials that are a little bit beyond what you would normally expect as far as a regular high school science teacher. Do you want to tell us a little bit about your background and why it would be appropriate for me to have this array of questions for you to answer today?
Okay, once upon a time I was a researcher. I have a PhD in plant science and a bachelor's in molecular biology, and many moons ago I did research in tomatoes, basil, ginger, and turmeric, and I did a lot of work with trying to transform sweet basil, transformation being putting foreign genes into sweet basil. And I also have done work in Arabidopsis, which is kind of a model system in plants, very easily transformed, also tobacco.
Okay. So with his background in those types of plants and the engineering, he and I have talked a little bit about Firefly Petunias. Now, his exposure to Firefly Petunias has been kind of through me and my work and my hobby that I've kind of created around them. My exposure to this idea actually came from a long time ago. So back in 1986, I believe is when it was, there was a plant that was created that had animal DNA that was inserted into a plant. So that was taking genetic material that made fireflies glow and putting that into a plant. In this case, it was tobacco, and that's where they created the firefly tobacco. And for myself as a kid growing up in a greenhouse and having a father who was an agronomist and really into plants, that really kind of spoke to me. And kind of from that moment on, after reading the article and learning about Firefly Petunia, I always wanted a plant that glowed. So when the Firefly Petunias came available last year, that to me was really kind of exciting. To be able to have something to basically hold in my hand that was a lab experiment that's basically being sold to the private consumer. And that's really kind of where we're at, is we're still working with a plant that is really basically what you would, we've talked a lot about this, but you would identify this as basically a lab specimen that is being released to the public, and it's not perfect. You would agree with that, right? Mm-hmm. So there is probably a lot of refinement that needs to go on over time, and there's a lot of issues that happen with these Firefly Petunias that we're going to kind of address some of those questions that people have had about those. Some of you watching are going to be a part of one of the Firefly community forums, so any of the Firefly Facebook groups. Some of you may even be folks that are hanging out on Reddit. I'm not there to join you yet. I have enough on my plate as far as answering questions off of YouTube and Facebook. But with this, I would probably say this is a very unstable plant. There's a lot of things that I've seen occur with this plant as far as mutations that are above and beyond what I would normally see as a nurseryman. And one of the problems that people are running into, and in fact, I've had it on this large specimen over here, is a point in the life of the plant where a portion of the plant just stops glowing. So if you're not familiar with Firefly Petunias, they're bioluminescent, they glow in the dark, and that's the whole premise behind them, is it's a plant that you can see in the dark because it lights up. But one of the problems we have is simply branches just turning off, and all of the glow goes, or it glows so faintly that it's really kind of hard to see. And that process is called gene silencing. Now, the reason I brought my science teacher in here to tell me a little bit about that is my knowledge of genetic engineering is old. It's really old. It's from the 90s. So basically, it's like Stone Age, knocking rocks together to get DNA to get into the plant. So that's why I'm tapping someone that has a little bit better knowledge than me. You know, the best thing that you can do when you don't have knowledge is find someone who does. So would you like to tell us a little bit about gene silencing, kind of what it is, how it works? You can go as technical or as basic as you want, but kind of explain gene silencing from the aspect of Firefly Petunias.
Okay, so gene silencing occurs in any type of biological system outside of a virus. Basically, what's going on is you overproduce, you take a gene, you produce an RNA from it, and then the RNA can, if it makes a hairpin, which is a double-stranded RNA, it's recognized by the cell as being abnormal, it's chopped up, and those pieces that are left can bind complementary pieces of RNA and tag them for destruction as well. And they can also go back into the nucleus, bind the complementary DNA, and target it for methylation, which basically turns off the gene. And gene silencing occurs naturally in the presence of viruses because you don't, normally in a cell you do not see hairpin RNAs unless there's a virus present. So a virus injects its genetic material into the cell and it, one way or the other, makes a RNA hairpin, and that's destroyed, and then it goes on to tag more viral RNA for destruction. And then if you have similar sequences in the DNA, it can tag them for methylation. And so it's often common that if you have overexpression of a gene in a cell, you can get RNA silencing as well. It eventually forms a hairpin, it's broken up, and then that tags other complementary RNAs for destruction and then methylation.
So this process is I think you earlier likened it to like an immune system.
It's the immune system.
So it's an immune system response to an error that's occurred. So it's a natural process to eliminate an unnatural occurrence. Mm-hmm. Okay. So with that, and I'm going to kind of seek some clarifying questions to make sure that myself and my viewers are all kind of on board. With this, it's simply not just going in and turning off that gene. It's not flipping a switch and leaving that gene intact. It's actually physically going through and removing and destroying it. Is that correct?
Yeah, you have the small piece of RNA binding to a viral piece of RNA or a overexpressed piece of RNA in the cell, which then produces the double-stranded RNA molecule, which your cell does not like. It targets it, and then once it does, you get that double-stranded RNA, it's destroyed. The pieces are left to bind to other complementary RNAs or go back into the nucleus for methylation of the DNA.
Okay. So, short answer is, once this occurs, there's no going back.
No going back.
Okay, so it's done. So when you have an individual that's got a Firefly Petunia and a portion of that has gone silent, the only way to fix it is to eliminate that tissue and just start over with something else or selecting another branch. Mm-hmm. Okay. So with this, we had some other questions that I had our Facebook group kind of pose. And with this, one of the questions was, why is the inheritance of gene silencing, why is it kind of spotty? Why does it occur in some plants but not occur in others? Is that just because the nature of gene silencing? Is it something that could be expected? Would there be, basically you're asking, is there a cause to what's triggering the gene silencing?
The cause could be whether or not, the cause could be a bunch of things, and the easiest way to do it would be to do a PCR test to see if it's present. If it's present and you don't see any phenotype, any glowing, then you know it's been silenced. Now, the more copies you have in the genome of your Firefly gene, the more likely it is to be silenced. If I remember right, the Firefly gene is being run by a 35S promoter, which is a viral promoter, which basically says, okay, anything under control of this promoter is drastically upregulated. So I'm getting huge amounts of Firefly transcripts, which then are turned into protein. And so the more transcript I have, the more likely I am to get a double-stranded piece of RNA if they're complementary, and then, which is then starts the silencing process.
Okay, so it's and as far as kind of our community's understanding with the Firefly Petunia, that there's multiple copies, that the goal is to increase the glow as much as they can, but from what I'm interpreting from what you're saying is the more we try to increase that glow, the more likely we are going to have silencing occur.
That'd be my guess.
Okay. So with the gene silencing, one of the things folks have questions about is if it's occurring in the plant and this is a known issue, and we'll just have you kind of theoretically propose some solutions or some thoughts that you might have, just kind of from your experience. What would be one of the best ways to prevent gene silencing?
Yeah, one way to prevent gene silencing, and this is just, I guess, speculative, would be put the Firefly gene under the control of a promoter that you can induce.
Okay.
So like you spray a chemical on the flower, it causes the Firefly gene now to be expressed.
Okay.
And then eventually when you're tired, when you don't want to see it anymore, you don't give it the chemical, so it's turned off.
Okay. So I think that's like, I think they actually did that.
Oh.
I think that was one of the things that they had done. Because I remember vaguely reading something about that, and you kind of reminded me of that. I think that was one of the approaches, but the group that was working on it, the feedback that they'd received is people didn't want to add anything to the petunia to make it glow. They wanted it to occur, you could say naturally in quotes, you know, it's not natural for it to glow, but
they want it to always be on.
Always be on, right. And I know that originally from my memory with the Firefly tobacco, when that came out, that it didn't always glow, it tended to glow when it was watered. So there was some sort of trigger that then made that cascade effect occur and the plant began to glow. So using a promoter, so a spray of some kind, some sort of chemical or that we're adding in would then make it glow better.
Yeah.
So it'd be more like a glow stick. You want to see it glow, you spritz it, it glows. As that chemical reduces, then it would fade.
Another way would just be to make the Firefly Petunia with other transformation events, and you might get it in a genome, part of the genome that is differentially expressed. So when you're talking about the bacteria, the tobacco, when it was under water stress, it turned on or turned off. Basically, it was the gene, despite they put it underneath an inducible promoter, it was under the control of another promoter that was involved in those processes.
Okay.
It's where that gene pops into the genome is fairly random, and they can have effects.
Okay. So with that, to stabilize it, kind of what you're saying is getting it into the exact right location that you want it is probably what would make it stabilize the best.
And good luck.
Okay.
That's not an easy process.
It's inserting genes with a shotgun, right?
Yes.
Pretty much. Okay. So with this, the implication would be that the glow genes landed somewhere in the genome. They're expressed, they're glowing, they may not be where we want them, but they're working. So kind of like the old pickup truck on the farm, if it still works, go with it. Don't try to fix it. You'll just make it work or make it not work. And then we had a note about, you remember this one about classical crossing? Was that to figure out where it landed or whether or not it was being expressed?
Yeah, so you would have to do a PCR test to see how many copies are actually in the plant and where they are. Because this could also affect, how many, the ratios of doing a crossing. If you've got two copies in the gene, unless they're on the same chromosome, it's highly unlikely that they're going to segregate together. If they're on different ones, they're more unlikely to stay together.
Okay.
So, yeah, you're gonna have to do a PCR test or do some laborious crossing experiments to figure out with other markers to see where they are, which chromosomes they are.
Okay, so we've talked a little bit about the gene silencing and kind of how that works, and really where that's going to be throwing up problems as far as the home consumer of fireflies. Basically, there's nothing we can do on our end. You know, there's not anything where we could go through and turn the switch back on. It sounds like, in a way, even breeding back to the original, it's not going to turn on the genes that have turned off. What it's going to do simply is just allow that plant the opportunity to maybe inherit both copies again. So one of the things that I had as an experiment, Firefly Petunias are white. They're white on purpose so that you can see the majority of the light that comes out of the blossom. And the blossoms, so anywhere where the tissue is rapidly growing is what glows most intently. All blossoms will glow, but the newest ones glow the most. So a white petunia, in my understanding or my assumption would be, was chosen so that you could see most of the glow in that. You know, if you have a dark purple petunia, you're not going to see it glow. So for me, white petunias were fairly plain, and I live in a nurseryman's world where I love things of color that adds more variety to a garden. It's not as monotonous as just plain white. So I crossed my Firefly Petunia with some just off-the-shelf store petunias. And the thing that was a surprise is, out of my 120 seedlings, all 120 seedlings did glow to some effect. Now, I will be honest and say that they're not as bright and as intense as the original Fireflies, but they do all, in fact, glow. There was a great online debate about this problem of having seedlings that glowed, and this is actually a cutting of that plant. It's not actually one of the glowing ones, or the crosses. But the real question was, how in the world, with everything we know from Gregor Mendel and genetics, what was it that I did without knowing it that allowed 100% of my petunias to glow?
So one explanation of getting 100% to glow would be if your gene, let's just say we're talking about one gene. If you've got two inbred strains, so you've got your inbred off-the-shelf strain and you've got your inbred Firefly strain, the Firefly, it's inbred, so by definition, it's homozygous. The same thing going for your off-the-shelf, that's inbred, so it's homozygous. If you cross them, you should, by definition, get a heterozygote, which would have at least one copy of the Firefly gene.
Okay. So simply, my opportunity was the chance of the seeds that I used.
Yeah.
So I used two inbred sets of seed. And these were, I think they came from Burpee. It was just an off-the-shelf one. I'll link the video where I showed the seed packet of where they came from. But what it appears is that they're both inbred, and then in combining together, we're getting at least one copy, because as that genetic material transfers in, it's dragging at least one copy into the plant. So therefore, if it's got at least one copy, it's going to glow at least some point. And that's probably most indicative of the fact that they're about, not quite, maybe a little bit more than half as bright as a standard Firefly Petunia. So that would lead us to believe that it's not being expressed as much because there's only a single copy. Mm-hmm. Now, with my Firefly Petunias, I've started a breeding program, and one of my goals is to bring in color, but also bring the brightness back. So now that I'm going to go back in and I'm going to cross my F1 generation, my first generation of hybrids, back with the original Firefly Petunia, is this the point where I'm going to start to see that differentiation of that percentage that are glowing as bright as the original mother plant? Is that the chance that we're going to see that, kind of what Gregor Mendel would have predicted?
You should see more brighter plants.
Okay.
Yeah.
Okay.
Whether or not it's acting in if it's acting as a monohybrid cross or a dihybrid cross, whatever, that's a different story, but you should start seeing writer ones.
Okay.
If you get more than one copy, because let's just assume you're getting one copy. Yeah, a homozygous will have 2 copies if there's 1 copy, because you got 2 sets of chromosomes. Yeah.
So basically what it is I'm going to be shooting for that plant that is going to get the missing copy back. It's not necessarily if it gets 2 copies of the original, that's not doing us any good. It's got to be on that other location of that gene. So kind of from there, we're going to expect to see that variance or that percentage of plants that, we've got a quarter or a third of them that are as bright as they should be, and then it varies down from there. And then of course we're going to have some that are probably completely silenced after that. So that's kind of where I'm going with that. Now, this is not just a one-off breeding program, so I'm going to continue with this, but I'm probably going to see a greater separation between the numbers that glow and the numbers that don't glow as I move on, which is kind of what I'm expecting. So that's kind of the answer that we're looking for. Is it was simply luck that I grabbed the right seeds and put them together, and that's kind of the result of that, is getting 2 inbred strains together then allowed that to occur. All right, so on behalf of my Facebook group and the YouTube subscribers that I have, I'd like to thank you for coming and helping us kind of solve some of those issues and understand things a little bit better. And this is really something that you and I do professionally. We answer questions. We try to make complex things more simple to understand, and draw from our experience and our background to help others understand complex topics and understand kind of the why and the how. So thank you for coming in and being a part of this video. I'm sure this won't be the last one. We'll come up with something else. Those of you that subscribe to the channel, you might have seen some of the other things we've done. There's the ethanol lab that we've done. I think there's a couple others that we might have shot videos on. This is also our beekeeper, so we might do some videos on that. But if you have other questions, go ahead and drop them down in the comments, and maybe we'll shoot a follow-up video maybe in a few weeks or a couple of months, maybe near the end of the summer, to answer any other questions or things that might come up. But again, I'd like to thank you for joining me today. And anyway, I hope you learned something. I hope you enjoyed. Ag Teacher Thoughts out.